Hello, it's Dr. Panku. Here's the lecture I promised you. During this lecture, there will be 3 secret words that will be given, and then you'll go to Blackboard where you'll find a quiz, and all you'll do is enter these 3 words, and you'll get your 3 points for 3 points for the questions, and then 2 points for attendance. All right, so we're taking a look at the 2 main types of cells, and you have already learned a little bit about this, but we're going to review it here in Chapter 4. If you recall the cell theory from lab, it says that all organisms are composed of cells and that all cells come from pre-existing cells. Schleiden and Schwann, 2 scientists independently, made these observations, Schleiden with animal cells and Schleiden with plant cells and Schwann with animal cells. And they, after observing many specimens, came to the conclusion that all organisms are composed of cells. We know that all cells have a plasma membrane. So the plasma membrane, as you learned in a previous lecture, is a phospholipid bilayer. The plasma membrane is a phospholipid bilayer in all living organisms, and in fact, even viruses steal the plasma membrane to surround themselves from the host cells that they infect. Plasma membranes are selectively permeable. They're picky about what can come across. Inside of the cell, there's going to be a gooey material, which is primarily water. That is called the cytoplasm, and the cytoplasm is where chemical reactions occur. We'll also find in the cell genetic material, both RNA and DNA, and that's necessary for growth and reproduction. RNA and DNA is required, as we learned in Chapter 1, in the definition of life. Remember, viruses just have RNA or DNA, never both. So there are 2 main types of cells based on the organization of the genetic material, whether or not there is a nucleus with a membrane surrounding it or a chromosome of DNA without a membrane surrounding it. Those 2 cells are called prokaryotes and eukaryotes. Pro means pre and kary means nucleus. So a prokaryote has no membrane surrounding its nucleus. The eukaryote has a nucleus with a membrane surrounding it. The DNA is found in both organisms. As we learned in a previous lab, the eukaryotes evolved from the prokaryotes. We know that there are 2 domains in prokaryotic cell, bacteria and archaea. Prokaryotic cells generally are smaller. In next week's lab, you will be seeing these prokaryotic cells. They are quite small and they're much simpler in structure, and this allows them to reproduce very quickly and very effectively. And of course, they reproduce asexually. This is just by splitting in half. This is an extremely successful group of organisms. It's probably been around for close to 4 billion years. The bacteria, of course, are prokaryotes, and they are well known because some cause disease. We think about bacteria as being bad, but really only 1% of all known bacteria actually cause disease. 99% of them have other roles in the environment. Some of them are even used to manufacture chemicals, foods, and drugs, and other things. For example, all of the vitamins that you take, if you take a vitamin pill in the morning, all of those vitamins are actually made by bacteria in an industrial process. Certain antibiotics actually are made by bacteria, and the bacteria make these to combat other bacteria. We just got lucky that somebody found them and put them in pill form, and we can take them when we have an infection. Bacterial structure involves a cytoplasm. The cytoplasm is primarily water surrounded by a plasma membrane and a cell wall. Now, this is one thing that's unique about bacteria. They have a cell wall that contains something called peptidoglycan. Peptidoglycan is a molecule that contains a carbohydrate as well as a protein, peptidoglycan. Sometimes there's a capsule surrounding the bacteria. And in fact, if they do have a capsule, they are more virulent, more likely to cause an infection. That's because our white blood cells try to gobble them up, but they slip off of that gooey capsule. So our first word that's going to be part of your quiz is capsule. Now, bacteria have a cell wall, and cell walls are necessary to maintain the shape of a cell. Bacteria come in 3 basic shapes: a sphere, which is called a coccus, a rod shape, which is called a bacillus, and a spiral shape, which is called a spirillum. So typically, these type of bacteria have names. Streptococcus, for example. If you have strep throat, you are infected with a bacterium that has a streptococcus shape. That's a spherical shape in chains. The cell wall is giving the shape to that organism. Bacteria also have DNA, but they don't have a nucleus. They have a single circular coiled chromosome located in the region called the nucleoid. So they only have one chromosome. It is double-stranded DNA, but it is circular, rather unusual. We have 46 linear chromosomes. Bacteria only have one circular chromosome. Inside of bacteria, we also see ribosomes. Ribosomes are the site of protein synthesis. Now, eukaryotic cells also have ribosomes, but the ribosomes are a little bit different in size. But that's all. Ribosomes are basically the same between prokaryotes and eukaryotes. Some of the antibiotics that we take against bacteria affect only bacterial ribosomes, and that's why we can take them. They don't hurt our ribosomes. Bacteria also may be able to get around. The appendages that they have are either flagella, which are long and thin, and they're involved in propelling the organism. In fact, some bacteria are quite good swimmers. There's one that causes a urinary tract infection called Proteus that actually can swim against the flow of urine while you're peeing. Some bacteria also have short bristle-like structures called fimbriae. Fimbriae allow these bacteria to stick to surfaces, like bacteria that are sticking to your teeth, making that biofilm you feel in the morning when you get up. These bacteria have these short bristle-like structures called fimbriae that allow them to attach to your teeth. So here we see the structures. We see the fimbriae, these short bristle-like structures that are involved in attachment. Then we see the cell wall. There's a capsule actually around this bacterium, so this is this aggregation that you see around the cell wall. The cell wall here is in purple, and then below that you have a plasma membrane. So the plasma membrane is that phospholipid bilayer. Sorry about that. Then inside we have the cytoplasm. These red structures are the ribosomes. Here we see the single circular chromosome. It's quite large, so it has to coil up. Well, it's sort of like a very big rubber band that would coil up inside of the cell. And then finally, we see these long projections here. These are flagella. So this is a rod-shaped bacterium, which would be called a bacillus. E. coli is a rod-shaped bacterium. And these flagella are for propulsion. Not all bacteria will have fimbriae, and not all bacteria will have flagella. So these are very small in comparison to the eukaryotic cells. The eukaryotic cells include the protista, the fungi, the plants, and animals. So everything that's in the domain eukarya that we learned before, those will be eukaryotes. That's easy to remember because eukarya, it sounds like eukaryotes. Eukaryotic cells have a membrane-bound nucleus that houses the DNA. They are much larger than prokaryotic cells, although there are a few exceptions to the rule. Get used to it. In biology, you're always going to find an exception to the rule. These cells are what we call compartmentalized. In other words, they have compartments inside them, and that's why they can get larger, because specialized activities will go on in certain areas of the cell. There are organelles. Organelles means little organs. These are membrane-bound, and they're going to be found internally. The nucleus and the ribosomes fall in one category. The endomembrane system is another category. The energy-related organelles, and then finally the cytoskeleton. So the cytoskeleton, you can imagine like our bones will be our skeleton. Cyto means cell skeleton, and that will give the cell some shape. So this is the structure of a typical animal cell. And if I could relate this a bit to you about the size of the bacteria that we saw in the previous slide, bacteria would be a little bit smaller right here than what you see here. This is a mitochondria. Mitochondria actually evolved from bacteria. So bacteria would have about this size. You can see then that the eukaryotic cell is much larger. It's much more complicated. It has a membrane-bound nucleus, and it also has these endomembrane structures. So this is a membrane-bound structure. This is a membrane-bound structure. Okay. One thing to notice here about the animal cell, it has a plasma membrane, but it does not have a cell wall. Now this is a typical plant cell. Big difference here. A couple of really big differences is number one, the cell wall. Get my pointer back here. Here's the cell wall, okay, in green. It's not really green. Cell wall has cellulose in it. Another big difference here is this structure. This structure is a chloroplast. Chloroplast will be where photosynthesis happens. And then this structure, you just saw before, that's a mitochondria, and plants have mitochondria as well. And another big difference here is this big water balloon. This is called a central vacuole. Plant cells have central vacuoles that are filled with water, and oftentimes they're filled with pigments, and they can also be filled with defensive type chemicals as well. So we're going to look at the plant cell as well as the animal cell in this chapter. Let's start with the nucleus. The nucleus in the eukaryote is going to be bound by a membrane. It's a plasma membrane, but it's a double membrane. The nucleus stores genetic information. Inside of the nucleus, we will see, when the cell is not dividing, diffuse DNA, protein, and some RNA. This diffuse DNA is called chromatin. Chromatin looks very grainy. I'm going to be taking a look at our cheek cells in lab next week, and you'll see that the nucleus looks quite grainy. That's because the chromosomes are not condensed, and they're present as chromatin. So imagine you had a ball of yarn, and you just let that ball of yarn go, a skein of yarn, just let it fall down in a basket. That's what the chromatin would look like. It would look like a big pile of spaghetti. But prior to cell division, DNA compacts into chromosomes. Chromosome, that is our second secret word. Chromosome. So chromosomes, as we'll be learning later, are actually just very tightly coiled chromatin. And remember, we have 46 linear chromosomes. Eukaryotes have linear chromosomes. Bacteria have circular chromosomes. Inside of the nucleus, we find a nucleolus. A nucleolus sort of means little nucleus. And this is a special region, sort of looks like a golf ball inside of the cell, inside of the nucleus. And that's where ribosomal RNA is made. Ribosomal RNA makes up ribosomes. Ribosomes are the site of protein synthesis. So ribosomal RNA has to associate with protein, and then it makes up the structure that is known as the ribosome. The nuclear envelope is a membrane. It's made of a phospholipid bilayer, but it is a double membrane. It has pores in it. That's necessary because stuff has to get in and out pretty easily. All of the things needed to make DNA have to be able to get in. And then the RNA message that comes from the DNA in making protein has to get out. So it's very important that things are able to get into and out of the nucleus. That membrane, you'll find later, actually does break down as the cell is about to divide. So here we see the nucleus, almost looks like a golf ball here, and we have the double membrane. These are the pores. This is a freeze fracture scanning electron micrograph, which is really interesting. You can see all of these pores on it. You can also see the nucleolus. That's the site of ribosomal RNA synthesis. And then inside you see a nucleoplasm. Nucleoplasm is like cytoplasm, but instead it's in the nucleus. And here you see the chromatin. Chromatin here is very diffuse. Attached to this outer membrane of the nucleus, you can see something called the endoplasmic reticulum, and we'll be getting that here to that in a moment. Okay. The ribosomes are actually a stage upon which proteins will be synthesized. So remember the central dogma, DNA to RNA to proteins. So the protein is going to be reading a message that is made of RNA and synthesizing proteins. Now remember, we do find ribosomes in both prokaryotes and eukaryotes. They're just larger in eukaryotes, but they have the same function, protein synthesis. Ribosomes are made of two subunits, and the two subunits are apart from one another until it's time to make a protein. They're made of a mix of proteins and ribosomal RNA. they're going to get the instruction to make the amino acids. Again, remember, DNA to RNA to protein. A protein is a polypeptide. Ribosomes are going to receive the messenger RNA to make the amino acids in a polypeptide, and we'll be learning that in more detail at the end of the semester. In eukaryotes, some ribosomes are free in the cytoplasm, but many of them are attached to a structure that is called the endoplasmic reticulum. Reticulum means spotted. So here we see a very good example. Here's the nucleus. Inside, you see the DNA. The DNA serves as the template to make messenger RNA. There's messenger RNA leaving the nuclear pore. So it's produced in the nucleus. It moves through a nuclear pore and it goes into the cytoplasm. The messenger RNA meets up with the ribosome. There are two parts, so the two parts clamp down together on that messenger RNA. That's this teal color here. And it synthesizes a protein. The protein is shown as these purple spheres. Okay. Now, if the protein needs modification, it's going to be modified in the endoplasmic reticulum. So the ribosome is going to dock on to a spot on the endoplasmic reticulum, and this protein is going to be extruded into the lumen or the opening of the endoplasmic reticulum. And here you can see this happening. There's that polypeptide. The polypeptide is oozed out of here, and then it folds up. As you learn, proteins in their tertiary structure fold up in their native conformation or normal shape. So this protein now is going to move through the lumen of the endoplasmic reticulum, and modifications are going to happen to it. In the meantime, once the protein is synthesized, the two subunits of the ribosome are going to come apart. The messenger RNA is going to be released, and then it will be degraded in the cytoplasm of the cell. So its job is over. The protein is synthesized. It will be degraded. So you can see there is a very close relationship between the nucleus and the endoplasmic reticulum, because proteins will be modified in the endoplasmic reticulum. The endoplasmic reticulum is part of the endomembrane system. This consists of the nuclear envelope, membranes of the endoplasmic reticulum, we abbreviate that as ER, the Golgi apparatus or Golgi body, named after Dr. Golgi, and a number of vesicles. These are membrane-bound components in the cytoplasm. So this helps to compartmentalize the cell. It restricts certain reactions to specific regions, and this is the reason why the eukaryotic cell can be larger. The endoplasmic reticulum is also known to release transport vesicles. The Golgi apparatus also can carry molecules from one part of the system to the other. So the endoplasmic reticulum is a complicated system of membranous channels and sacules. It is continuous, so it begins at the nuclear membrane and it winds throughout the cell. A good part of the cell is going to be taken up by the endoplasmic reticulum. There are two types of endoplasmic reticula that we find in the cell, and how much of one each cell has depends upon the function of that cell. The rough endoplasmic reticulum is called that because it looked rough when scientists first observed it with an electron microscope. They found endoplasmic reticula that looked rough and others that looked smooth, so that's why they called it that. Later it was found that the reason it was rough was because the ribosomes were attached to it. Remember, those proteins are extruded into the lumen of the endoplasmic reticulum, so the rough ER is involved in protein modification. Proteins that are extruded into the endoplasmic reticulum are going to be modified to a certain extent. The rough endoplasmic reticulum then will form little blebs or bubbles that are going to go to the Golgi apparatus. These are called transport vesicles. So you can imagine little bubbles coming off, and the bubbles are going to be transported to the Golgi with the help of the cytoskeleton that we'll learn about momentarily. The smooth endoplasmic reticulum is continuous with the rough endoplasmic reticulum but has no ribosomes. So the function of the smooth ER is to make lipids. like phospholipids and steroids. So you know phospholipids are found in the cell membrane, steroids are hormones. So the function depends on the cell. For example, if the cell is responsible, we're looking at pancreatic cell, let's say, is responsible for making insulin, we would find a lot of rough endoplasmic reticulum because insulin is a protein hormone. On the other hand, if we're looking at a testicular cell that is responsible for making testosterone, we would find more smooth endoplasmic reticulum because testosterone is a lipid hormone. It's not a protein. So here you see the endoplasmic reticulum. Here is rough ER. You can see all the little dots on there. So the rough ER has the dots, the smooth ER does not. And you can see how almost like a maze the endoplasmic reticulum is. It is continuous with the nuclear envelope. The Golgi apparatus is a stack of flattened vacuoles. It's almost like pancakes, a stack of pancakes, but they're hollow inside. The Golgi apparatus is very much like a transfer station. So it's going to take these bubbles, the vesicles from the endoplasmic reticulum, and then those bubbles are going to fuse with the Golgi apparatus vacuole. If you have two soap bubbles that come together, they will form one soap bubble. And this is exactly the same idea that's happening here. The membrane structure is exactly the same. So when the endoplasmic reticulum buds off a bubble, which is called a vesicle, the Golgi apparatus can receive it. And so whatever is inside of that whatever is inside of that vesicle is going to dump into the Golgi apparatus. It modifies molecules then within the vesicles. That's the role of the Golgi apparatus. It sorts, it repackages whatever is in there for a new destination. So it's sort of like the barcoding station at UPS. So it is going to mark for delivery the molecules that are going to be released. Some are going to stay within the cell. Others are going to be exported out of the cell. Inside of the cell, we also find vesicles that are called lysosomes. Lysosomes are vesicles that bud from the Golgi apparatus, and they stay in the cytoplasm. They've got some pretty nasty things inside of them. So sometimes they're called suicide sacs, simply because if they break open, they would destroy the cell from the inside out. Their role is to get rid of worn out cell components and also to gobble up disease-causing organisms. So we find a lot of lysosomes in the cells that are involved in our immune response, like white blood cells. When you get a cut, white blood cells are going to leave your bloodstream, go to the site of the cut, and attempt to gobble up these bacteria. Now, if the bacteria have a capsule, it's a little bit more difficult for them. But regardless, the white blood cells are going to just gobble up via a process called phagocytosis all of these bacteria. They will die in the process. And afterwards, what you typically will see is pus. Pus is just an aggregation of dead bacteria and dead white blood cells. So inside of the lysosomes, we find these degradative enzymes, and their job is to degrade whatever comes into the cell through phagocytosis. We'll learn more about phagocytosis in Chapter 5. So here is the entire endomembrane structure in its glory. We have the outer membrane of the nucleus, that double membrane structure, the rough endoplasmic reticulum. Here we have the smooth endoplasmic reticulum and the Golgi apparatus. So here you can see these vesicles budding off. They bud off of the rough endoplasmic reticulum. They also may do it from the smooth endoplasmic reticulum. Sometimes they'll stay in the cytoplasm, but other times they are transported to the Golgi. Here you can see this vesicle that is fusing with the Golgi's membrane, and in the membrane then there's going to be some modification of the molecules, and then they are going to leave. Okay? When they leave, then they can perhaps be lysosomes. In this case, we see a lysosome that is fusing with what's called a phagosome in phagocytosis. This is phagocytosis. So this vesicle is bringing, let's say these are strep bacteria coming into the cell. They are inside of a vesicle. Okay, that's called a phagosome. The phagosome fuses with the lysosome to make what's called a phagolysosome. Don't worry about these words right now. We'll get to them later. This is a phagolysosome, and in here, all the chemicals are going to degrade these bacteria to kill them. Okay, let's see if we can watch this. Might not be able to. Let's see.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
Okay. Then there are vacuoles inside of the cell. Plants, remember that great big water balloon I showed you? Plants have vacuoles in them. These are membranous sacs and they are larger than vesicles. Vacuoles can be used to rid a cell of excess water. These are called contractile vacuoles. And in lab, hopefully we will see a paramecium. Paramecium gets rid of the excess water by a contractile vacuole. Sort of acts like the spout of a whale if you think about it. Vacuoles can also be involved in digestion of intracellular material and also for storage. For example, pigments in plants we'll see in lab are very commonly inside of the vacuole. In us, we have cells called adipocytes. Adipocytes are going to store fat cells, and we find them in adipose tissue. So here you can see some great examples. This is paramecium right here. Now this is an electron micrograph. So this is magnified 800 times. We're only going to magnify ours about 100 times to see them. But you can see these contractile vacuoles here. Almost looks like an anus, and it's going to squirt out extra water that is in the cell. Then here you can see human fat tissue, and these cells right here are called adipocytes. And in these vacuoles, we store fat, and they can get bigger and bigger and bigger, just storing lots and lots of fat. And this is an electron micrograph of a plant cell. This plant cell here, we can see the chloroplast, and there's that big water balloon that's in the center of the plant cell. So it gives rigidity to the plant cell, allows it to stand up tall when there's plenty of water in there, and sometimes there are chemicals in there that are involved in defense for the plant, and other times there are pigments in the vacuoles. We also have energy-related organelles, two of them. Plants have chloroplasts. Chloroplasts are the site of photosynthesis, so they use solar energy to synthesize carbohydrates through the process of photosynthesis. We find chloroplasts in plants and algae. Okay. So typically chloroplasts are green. They have a three-membrane system, a double membrane enclosing a liquid center. The liquid center is called the stroma. Thylakoids form the third membrane, and the thylakoid membrane contains pigments that capture solar energy. Those pigments include chlorophyll, but not limited to chlorophyll, as we'll learn in Chapter 8. Chloroplasts have their own DNA and ribosomes. Now, what's interesting about this DNA and their ribosomes is that the DNA is circular and the ribosomes are small. Remember that plants have linear DNA and larger ribosomes in their cytoplasm. So this gives credence that eukaryotes evolved from the prokaryotes because chloroplasts are like bacteria. They actually have bacterial DNA, and their ribosomes are much more prokaryote-like than eukaryote-like. This theory is called the endosymbiont theory and was proposed by Lynn Margulis, the late Lynn Margulis, about, must have been about 40 years ago. So here we see that triple membrane system. We have this double outer membrane here. get my pointer, the double outer membrane right here, and then this is considered the third membrane. This is the thylakoid membrane. The thylakoid membrane surrounds the thylakoids, so they sort of look like green Mentos, and they're stacked on top of one another. So this is the chloroplast here. And in lab, you saw the elodia, and you saw these chloroplasts in there. They look like little green balls inside of the, you saw the chloroplast inside of the cell. Okay, so a little close up here, some names to be familiar with. We have the double membrane, the outer membrane and the inner membrane. Then we have these stacks of Mentos. They are called thylakoids. Thylakoids are hollow. Well, not really hollow. There's a liquid inside of there, but there is a space. Okay, so that's called our thylakoid space. And then there is the thylakoid membrane. Now, surrounding all of this stuff is a gooey liquid, and this gooey liquid is called the stroma. Okay, a lot of terms to remember here, so hang in there. So we've got the stroma surrounding the thylakoids, and the thylakoids stacked on top of one another are called grana. Okay, this is one granum. There are many grana inside of the cell. So I tend to think of silos storing grain. So this looks like a silo to me, and I remember the word granum. Now, chloroplasts are a lot like mitochondria because they are also membranous structures. Mitochondria are found in both plants and animals, and that's important to remember. When I was a student, I thought plants had chloroplasts and animal cells have mitochondria, and I forgot that animal cells have to make ATP also. They need energy. So mitochondria is responsible for making ATP, which is cellular energy, and they're found in both plants and animals. Usually we can only see these under an electron microscope. They are quite small. They have a double membrane as well, and their whole purpose is to make adenosine triphosphate. The process that makes adenosine triphosphate is known as cellular respiration. Cellular respiration is the reason that we breathe. Cellular respiration needs oxygen. The process produces ATP, and carbon dioxide is a waste product. So we exhale carbon dioxide. Now, a few different terms, unfortunately, here with mitochondria. The inner membrane folds are called cristae, right? So remember that the inner membranes are called thylakoid membranes in plants. In the chloroplast, the mitochondria, they are called cristae. Remember in biology, every time we see folding, we know that increases the surface area. Increasing the surface area gives more surface area for things to react. Then the gooey liquid that's inside the mitochondria is called the matrix. In the chloroplast, it's called the stroma. In the mitochondria, it's called the matrix. The matrix is made of a mixture of enzymes, and they are going to be involved in breaking down carbohydrates to make energy, and the reactions that are involved in cellular respiration make ATP. Now guess what? The matrix also contains its own DNA and its own ribosomes, and they're more like bacterial DNA and ribosomes than the eukaryotes. Okay. So again, evidence that the eukaryote evolved from the prokaryote. So here we have an image shows us the matrix. So this would be a gooey liquid inside of here. Sorry about that. We have the inner membrane, the outer membrane, so we have that double membrane system. And these folds, which you can see on this electron micrograph, are called cristae. So on the cristae will be the respiratory enzymes that are involved in making ATP. So this is a great magnification here of 70,000x. Okay. The last thing I'd like to talk about is the cytoskeleton and motor proteins. The cytoskeleton is a network of interconnected protein filaments and tubules. So it's like the skeleton inside of the cell, but it's not made of bones. It goes all the way from the nucleus to the plasma membrane, and we only find it in eukaryotes, although actin, which is a component in eukaryotes, has also been found in prokaryotes. It is responsible for maintaining the cell shape. So imagine a cell that does not have a cell wall. It would easily collapse. But with the cytoskeleton, organelles are going to be held in place, and it's also going to get a nice shape. Imagine if you didn't have a cytoskeleton, every time you stood up, your nuclei would fall to the bottom of your cells, but the cytoskeleton actually holds them in place. It's also an internal highway. You can sometimes see chloroplasts actually moving around in a plant cell on this internal cytoskeletal highway. It's made of 3 types of filaments. There's a large, a medium, and a small. The large is the microtubule, the medium is the intermediate filament, and the small is the actin filament. Microtubules are small, hollow cylinders, so they're like rigatoni. They're assembled by a part of the cell called the centrosome, and they help to maintain the cell shape, and they're nice highways for organelles and other materials to move. The ones that are intermediate in size are called intermediate filaments. They're more like a rope, and they run from the nuclear envelope to the plasma membrane. So here you see the microtubules. Microtubules are made of a protein called tubulin. Tubulin is a spherical protein, so it's almost like a string of pearls on a necklace, and it's wound around and around and around. It's hollow in the center, so it's like a big rigatoni. It helps to maintain the cell shape, and it acts as a track for organelles and other materials to move. So you can see it extends throughout the entire cell, and it arises from the centrosome. The intermediate filaments are rope-like in structure, as I mentioned. And then there are actin filaments. Actin filaments are much smaller, as you can see here. They have 2 chains twisted into a helix. So imagine 2 strands of pearls just twisted. And this forms a dense web. It's around the outside of the cell and also goes up into any extensions. These extensions you see here are found in our intestines. These are called microvilli. So once again, folded, folded, folded. All this folding increases the surface area, which allows our intestines to absorb a great deal of nutrients. It also supports those microvilli, so they don't flop around. It's almost like having a lot of hairspray on your hair. It's going to stand up really tall, and that's going to increase the surface area. So actin filaments are also found in other cells that help the cells to move. Okay, motor proteins are instrumental in allowing cellular movement. There's one called myosin. Myosin interacts with actin, and that's how our muscles move. So cells as white blood cells or amoeba. You saw amoeba moving in lab. They will, I'm sorry, you're going to see that next week. That will allow the amoeba to move in an amoeboid fashion. So myosin interacting with actin allows amoeba to move, allows white blood cells to move, and also allows your muscles to move. 2 other motor proteins are called kinesin and dynin. Kinesin and dynin actually are motor molecules that walk, pretty much like literally walk across the microtubules of the cytoskeleton. There are receptors, and they're going to move across the cytoskeleton carrying things. They can carry transport vesicles from the Golgi apparatus to the final destination. I have a movie here with myosin interacting with actin. Let's see if we can get to it. Nope, sorry, can't get to that one. Up. All right, I will try to show that to you in lecture when we see each other next. So here we have kinesin and dynin, and this is an example of kinesin and dynin. Okay, so here's kinesin. Kinesin is a molecule that has these 2 stick-like leg structures, and they have receptors. The receptors attach to the microtubule, and they actually walk along here carrying something. Now here's a vesicle. Let's say we have a vesicle that needs to go from the endoplasmic reticulum to the Golgi apparatus. So it's going to be picked up by a kinesin molecule, and with the help of ATP, because it'll need energy to walk along here, it will walk along this microtubule and deliver the vesicle to the Golgi apparatus. So our last word is kinesin, K-I-N-E-S-I-N, kinesin. Okay, so that's the last of it. I'll pick up on with centrioles when I come back on Monday, and you can go onto Blackboard now and you can find that quiz. You're going to click on the quiz. You'll simply add these 3 words that I told you. Say them real fast. capsule chromosome and kinesin, and you can enter those and you'll get your 3 points plus your 2 attendance points. Have a great weekend.